Imagine a laboratory mouse inside um, a standard plexiglass enclosure. By all accounts, it's a completely docile, normal male mouse.
Right, just minding its own business.
Exactly, sniffing around the cedar shavings, totally calm. But protruding from the top of its skull is this tiny, incredibly thin fiber optic cable.
Which is hooked up to a laser outside the cage.
Yeah. So a researcher reaches over to a dial on a machine and turns on that laser, sending a specific frequency of light down the cable directly into one tiny microscopic cluster of neurons deep inside the mouse's brain.
And the change is instant.
It is. The mouse's entire demeanor it changes completely.
Hmm.
The researcher drops an inflated rubber medical glove into the cage. Normally a mouse would, ignore this or run away.
Sure, it's not a natural threat.
Right. But this mouse lunges at the rubber glove and violently, viciously attacks it. No conscious thought, no provocation, just pure, hardwired aggression unleashed by the flick of a switch.
It's a striking visual.
It really is. We are talking about the biological master switch that controls everything from your darkest, most violent impulses to your most basic life or death need for a glass of water. So welcome back to our deep dive.
Glad to be back.
If you're joining us right now, you are stepping into the second half of our exhaustive exploration into Chapter 41 of Principles of Neural Science.
That's right. The hypothalamus. Autonomic, hormonal, and behavioral control of survival.
And just a reminder, you are in an advanced neuroscience masterclass right now. We are not skimming the surface today. We're going straight to the depths of the biological machinery.
Yeah, in the first half of this analysis, we spent a significant amount of time establishing the hypothalamus as the master coordinator of homeostasis.
Right, the grand integrator.
Exactly. We mapped out how it receives all this interoceptive sensory feedback. The constant internal status report from every organ in the body. We looked at the sensors. Now we're pivoting to the effector systems. The output.
I really want to focus on that transition from sensing to doing. We know the brain is monitoring everything, right? Like blood pressure, temperature, nutrient level.
It's constantly monitoring. Yeah.
But I'm trying to picture how it actually reaches out and alters the body. Like, how does it physically manifest a change in your stomach or your kidneys or your heart?
That is exactly the mission for this half of the deep dive.
Perfect. So we're going to explore the top-down autonomic control, the massive neuroendocrine chemical broadcast system, the deeply complex homeostatic circuits for temperature, water, and energy.
And finally, those hardwired circuits for behaviors like sex, aggression, and parenting.
Okay, let's unpack this, because in the first half, we talked about reflexes happening down in the brainstem. But the brainstem doesn't always have the final say, does it?
Oh, far from it. I mean, the foundational brainstem reflexes handle the day-to-day, moment-to-moment automation.
Like breathing.
Right, your breathing rate adjusts, your blood pressure stabilizes when you stand up, all handled at a lower level of the central nervous system.
So it's background processing.
Exactly. But there are moments, usually extreme high-stakes situations, where higher brain regions just have to step in and completely commandeer that autonomic system.
An override.
A systemic executive override, yeah.
Hmm.
And one of those critical structures for executing this override is the periaqueductal gray, or the PEG.
Let's locate this for a second. The PEG is a region surrounding the cerebral aqueduct, right? That fluid-filled canal running right through the midbrain.
You've got it. It sits perfectly between the higher cortical areas and the lower brainstem.
So it's a bridge.
Its location is central to its function. The PEG receives inputs from almost every part of what we call the central autonomic network, meaning the cortex, the amygdala, the hypothalamus.
It's like a massive integration hub.
Exactly. When the brain detects a situation requiring an integrated, explosive behavioral and autonomic response, the classic fight or flight scenario.
Right, the panic button.
Yeah, the PEG is what projects down to the medullary reticular formation in the brainstem to actually make it happen.
I really want to visualize the mechanics of this, because when we hear fight or flight, we just think of like a racing heart or suddenly sweating. It feels like a generalized panic.
It feels chaotic, but biologically it's highly structured.
Right. At the microscopic level of blood vessels, what is the PEG actually doing to the architecture of the circulatory system?
It is actively and aggressively redirecting the flow of physical resources. It isn't just turning everything up blindly.
It's being selective.
Highly selective. During a severe threat, the PEG initiates a mechanical shift in blood flow. It sends descending signals down to the sympathetic preganglionic neurons in the spinal cord.
And those control the blood vessels.
Yes, they directly control the vascular, smooth muscle, the muscular lining of your blood vessels. The result is a massive, immediate vasoconstriction in the digestive system.
Wow. So it literally clamps down on the blood vessels surrounding your stomach and intestines.
It squeezes them completely shut. It reduces the blood flow to the gut to an absolute minimum.
Where does all that blood go?
Well, simultaneously, it sends signals to promote profound vasodilation widening of the blood vessels in the skeletal muscles, specifically the hind limbs of an animal, or your legs.
That's incredible. It's actively starving the digestive tract of oxygen and nutrients just to supercharge the legs for running away?
Because digestion is a biological luxury. I mean, you can't afford to expend energy breaking down a meal if you are about to become a meal for something else.
That puts it in perspective, yeah.
So the PEG orchestrates this instantaneous, massive redistribution of cardiac output so that the muscles have maximum oxygen to perform explosive movements.
But the PEG isn't acting alone in this override system, right? The source text brings in the medial prefrontal cortex, the MPFC.
Yes, the MPSC is crucial here.
And I always struggle with this a bit because we're trained to think of the prefrontal cortex as the seat of high-level human cognition, like complex thought, abstract decision-making, planning for the future.
The quintessential human traits, right?
Yeah. So what is it doing tangling with basic autonomic functions like blood pressure?
Well, we really have to expand our view of the prefrontal cortex. Yes, it handles executive function, but specific subregions of it have a profound visceral sensory motor component.
Okay, so which subregions are we talking about?
We're looking specifically at the rostral insular cortex and the rostromedial tip of the cingulate gyrus.
Which includes the infralimbic and prelimbic areas.
Exactly. This is the exact anatomical intersection where higher cognitive processing meets raw, physical bodily function.
So thought meets flesh.
Literally.
Yeah.
If you take an electrode and stimulate these specific areas in the MPFC, you don't just alter an animal's thought process or attention.
What happens?
You elicit immediate physical autonomic effects. You can induce sudden contractions of the stomach muscles. You can cause immediate dramatic spikes or drops in blood pressure.
It makes me think of an analogy. Tell me if this works. Imagine the autonomic nervous system is this massive global manufacturing company.
Okay, I like where this is going.
The brainstem and the spinal cord, they're the local factory floor managers. They run the day-to-day operations perfectly. They keep the machines humming, manage the supply chain, adjust the thermostat so the workers don't overheat.
Very steady, very routine.
Right. But the MPFC and the PEG, they are the corporate executives sitting in a glass boardroom at the very top of the skyscraper. Most of the time, they don't care about the factory floor. They just let it run itself.
They're focused on the big picture.
Exactly. But if there is a sudden existential crisis, like a massive market crash, or in biological terms, a predator leaping out of the bushes.
The executives step in.
Yeah, they don't send a polite memo. They slam a big red button on their desk that sends emergency override codes directly to the factory floor.
And that forces the entire plant to instantly pivot production.
Right. Shut down the cafeteria, which is the digestive system, and route all electrical power to the defense systems.
That boardroom analogy captures the hierarchy perfectly. The descending projections from the MPFC and the PEG completely bypass the slow, routine, regulatory feedback loops.
They just force the issue.
They enforce an immediate systemic change based on high-level cognitive evaluation of a threat.
Mm-hmm.
And to complete that boardroom, we have to include the amygdala.
Oh, right, the amygdala.
Because the amygdala processes intense emotional salience and fear. It forms descending networks right alongside these cortical areas.
So it's adding the emotional weight?
Yes. It projects straight down to the hypothalamus and the lower brainstem to ensure that your physiological state instantly matches the emotional reality of the terror you're perceiving.
Okay, so all of that, the PEG, the MPFC, the amygdala, they're acting through the autonomic nervous system, the sympathetic and parasympathetic nerves projecting out to the organs. It's like a series of hardwired, high-speed fiber optic cables running from the brain to the tissue. But we really have to pivot now to the second effector arm of the hypothalamus.
The neuroendocrine system.
Yeah, because if the autonomic system is a point-to-point fiber optic cable, the neuroendocrine system is a chemical radio broadcast.
The distinction is vital. The autonomic system is fast, electrical, and highly targeted to specific organs.
But the neuroendocrine system takes a totally different approach.
It relies on the bloodstream. It dumps chemical messengers, hormones into the blood, which then wash over the entire body.
So any target organ anywhere can pick up the signal.
Exactly. And the central command station for this massive chemical broadcast is the pituitary gland, sitting in a tiny bony pocket right beneath the hypothalamus.
But before we get into the actual hormones, we have to establish a stark anatomical reality here, right? Because the pituitary is not just one single monolithic gland.
No, it's really not. It is two entirely different organs with entirely different embryological origins, completely different cellular structures that just happen to be mashed together into one structure.
Which is wild to think about. We have the posterior pituitary and the anterior pituitary.
Right.
Let's start with the posterior pituitary because anatomically, it is literally just a direct neural extension of the brain itself. It hangs down from the hypothalamus on a stalk.
Embryologically, it grows down from the neural tube. The posterior pituitary, also called the neural hypophysis, doesn't actually produce hormones itself.
Wait, really? I always thought it made hormones.
No, it just stores and releases them. It contains the axon terminals of neurons whose cell bodies sit way high up in the brain inside the hypothalamus.
So the cells are in the brain, but their wires reach all the way down into the gland.
Yes, specifically, these are the magnocellular neurons. Magno meaning large. These large cell bodies are clustered tightly in two specific locations, the paraventricular nucleus, or PVH, and the supraoptic nucleus.
And these massive neurons are famous for producing two very specific chemical messengers, vasopressin and oxytocin.
Correct. Vasopressin, which is frequently referred to as antidiuretic hormone, controls blood pressure and commands the kidneys to aggressively reabsorb water back into the blood rather than turning it into urine.
And then there's oxytocin.
Right. Oxytocin is best known for its role in reproduction and parenting, causing a powerful uterine contractions during childbirth and triggering the letdown reflex for milk release during lactation.
I want to slow down and look at the actual molecular mechanics of this because it is an incredible logistical feat for a single cell.
It's an enormous challenge, yeah.
Both vasopressin and oxytocin are tiny, right? They're 9 amino acid peptide hormones.
Very small. Structurally nearly identical, probably the result of some ancient evolutionary gene duplication millions of years ago.
But how does a neuron sitting up in the hypothalamus actually get this physical peptide down into the pituitary and out into the blood?
It requires a massive internal logistics operation. Like all peptide hormones, vasopressin and oxytocin are not simply synthesized as the final tiny 9 amino acid product.
They start bigger.
Much bigger. They are synthesized back in the cell body of the magnocellular neuron as much larger, bulky precursor molecules, which we call prohormones.
Okay, so they make the big prohormone first.
Yes, and once synthesized, the cell's Golgi apparatus packages these large prohormones into transport vesicles.
So they're basically stuffed into these microscopic spherical shipping containers inside the cell body.
Exactly. And those containers have to travel. These transport vesicles are physically carried down the incredible length of the axon,
Like cargo.
moving away from the cell body in the hypothalamus, traveling down the entire length of the pituitary stalk, and finally reaching the axon terminals resting in the posterior pituitary.
But wait, there's still prohormones at this point.
Here is the brilliant part.
Yeah.
The manufacturing doesn't stop during transport. Enzymes located inside the moving vesicle are actively cleaving the prohormone as it travels.
You mean they are chemically snipping away the excess protein sequence while it's en route?
Yes, exactly.
It's like a mobile assembly line. By the time the shipping container reaches its destination, the final product is ready.
By the time the vesicle arrives at the axon terminal, the prohormone has been fully processed into the mature, active 9 amino acid peptide. Now the terminal is just waiting.
Waiting for a signal.
Right. When the magnocellular neuron fires an action potential, the electrical signal reaches the terminal, causing those vesicles to fuse with the cell membrane.
And they dump their cargo.
They dump the vasopressin or oxytocin directly into a bed of fenestrated capillaries, blood vessels with tiny pores in them, allowing the hormone to instantly sweep into the general systemic circulation of the body.
Wow. So it's a direct neuronal data dump straight into the blood. That's the posterior side. But the anterior pituitary, the front half, operates on a completely different set of rules.
Completely different. The brain cannot just drop its axons in there.
Because it's not brain tissue.
It is not made of neural tissue at all. It originates embryologically from the roof of the mouth, actually, migrating upward during development to meet the brain.
That's so weird to think about.
It is. It is a non-neuronal glandular endocrine structure comprised of 5 distinct types of hormone-secreting cells. And because there are no neurons there, the brain has to use a chemical middleman to communicate with it.
And that middleman system relies on a different set of neurons back in the hypothalamus, right? The parvocellular neurons. Parvo meaning small.
The parvocellular neuroendocrine zone, yes. These neurons do not send their axons down the stalk into the anterior pituitary. Their axons are much shorter.
Where'd it go?
They terminate much higher up, at a structure right at the base of the brain called the median eminence. The median eminence is key because it contains a highly specialized capillary bed called the hypophyseal portal system.
I think we need to define a portal system for the listener, because normally an artery brings oxygenated blood to an organ, it breaks into a tiny capillary bed to exchange nutrients, and then it gathers into a vein that goes all the way back to the heart.
That's the standard plumbing, yeah.
But a portal vein breaks that rule.
It does. A portal vein connects two capillary beds directly to each other, without traveling back to the heart in between.
So it's a shortcut.
A very specific shortcut. The parvocellular neurons release their specific chemical signals, which we call releasing factors or inhibiting factors, into this first capillary bed at the median eminence.
Okay.
The portal veins then carry these factors straight down, just a few millimeters, into the second capillary bed located inside the anterior pituitary.
So it's a private, direct blood channel from the brain to the gland.
Exactly. The releasing factors diffuse out of the portal capillaries and bind to receptors on those endocrine cells in the anterior pituitary.
Telling them what to do.
This is the brain commanding those cells to either secrete or stop secreting their own specific massive hormones into the general circulation of the body.
I want to trace a few of these specific circuits because this is where the brain really takes the wheel of the body's entire hormonal landscape. I mean, I'm struggling to picture how many different systems this controls.
It's expansive. Let's look at a few examples, starting with reproduction. GnRH.
Gonadotropin-releasing hormone?
Right. Its anatomy is unique. The neurons that produce GnRH are not clustered tightly in one single nucleus like the magnocellular neurons are.
They're spread out.
They are atypically scattered in a loose continuum that stretches from the medial septum all the way through to the mediobasal hypothalamus.
And how do they know when to fire?
They don't fire spontaneously on their own schedule. They're heavily regulated by upstream neurons that release a peptide called kisspeptin.
Kisspeptin?
Yes. When kisspeptin triggers them, they release GnRH into the portal system. The anterior pituitary receives the GnRH and responds by releasing luteinizing hormone LH and follicle-stimulating hormone FSH into the whole body.
Which ultimately drive the function of the ovaries and testes.
Exactly. That's the reproductive axis.
And then there's the stress response. CRH, corticotropin-releasing hormone. The text frames this as the final common pathway for stress, which sounds incredibly dramatic.
It is dramatic. It is the biological funnel for all human stress. The CRH neurons are tightly packed in the paraventricular nucleus, the PVH.
Okay, so they're localized.
Very localized. Think about what stress actually is. It could be psychological worrying about a looming financial deadline. It could be physiological, a severe viral infection or a broken bone.
Or environmental, like freezing cold temperatures.
Exactly. All of these wildly different sensory and cognitive signals converge on these specific CRH neurons in the PVH. When activated, they release CRH into the portal system.
And the anterior pituitary reads that signal.
It receives it and responds by releasing ACTH, adrenocorticotropic hormone, into the general blood supply.
And ACTH is the messenger that travels all the way down to the adrenal glands, right?
Yes, it travels through the blood to the adrenal cortex, the glands sitting like little hats on top of your kidneys, and commands them to synthesize and release cortisol, the primary human stress hormone.
Wow. So that entire massive systemic cascade, which alters your immune system, your blood sugar, your memory consolidation.
All of it.
It's all initiated by that tiny cluster of parvocellular neurons in the PVH, releasing a microscopic amount of CRH.
It's an incredible chain reaction.
So we have GnRH for sex, CRH for stress. We also have TRH, thyrotropin-releasing hormone, which drives the thyroid system to regulate metabolism. But it isn't all gas pedals, right? The brain can hit the brakes on the pituitary too.
It has to. Homeostasis requires both excitation and inhibition. For instance, the hypothalamus releases somatostatin into the portal system, which acts as a powerful brake, inhibiting the anterior pituitary from releasing growth hormone.
It's always a balance.
Always balancing the accelerator and the brake. We also see this with the arcuate nucleus of the hypothalamus. It outputs GHRH, growth hormone releasing hormone, the accelerator for growth, but it also outputs dopamine,
Wait, dopamine.
Yeah, dopamine.
Because we always talk about dopamine in the context of reward addiction and motor control in the basal ganglia. What is it doing in the neuroendocrine system?
It's acting in a completely different capacity here. In this circuit, dopamine does not act as a classic neurotransmitter jumping across a microscopic synaptic cleft.
So what is it doing?
It acts as an inhibiting neurohormone. The arcuate neurons dump dopamine into the portal blood. It travels down to the anterior pituitary, and it actively suppresses the endocrine cells there from releasing prolactin.
When you lay the mechanics out like that, the biological elegance of this two-step releasing factor cortical system is just staggering to me.
It is remarkably efficient.
The textbook points out how it acts as a biological amplifier. The hypothalamus only needs to release a few nanograms of CRH. That tiny amount hits the anterior pituitary, triggering it to release micrograms of ACTH.
And then that travels to the massive adrenal glands.
Right, triggering them to release milligrams of cortisol. It's a cascading amplification that starts with a tiny cluster of neurons and ends up altering the metabolism of nearly every single cell in the human body.
It is the ultimate translation mechanism. It is how neural information, thoughts, perceptions, fears is literally translated into a systemic physiological reality. The brain perceives and the body physically transforms.
Which brings us perfectly to the core of this entire textbook chapter. Having mapped out these effector arms, the electrical autonomic override, and the chemical neuroendocrine broadcast, we now need to examine how the hypothalamus actually deploys these tools to solve specific life-or-death survival problems.
The real-world applications.
Exactly. This is the homeostatic masterclass. We are going to look at 3 dedicated hypothalamic systems: temperature, water, and energy. Let's start with thermoregulation, regulating body temperature. Why is the brain so obsessed with a few degrees of heat?
Because thermoregulation is a strict physical constraint on life. We are chemical engines.
Right.
If your core body temperature drops too low, the kinetic energy of your molecules drops, and basic enzymatic reactions slow down to a halt. You undergo metabolic failure.
And if it gets too hot?
If your core temperature goes too high, the physical structures of your proteins begin to unfold. They denature. To manage this incredibly narrow window of survival, the hypothalamus utilizes a specific integration center, a cluster of neurons located in the median preoptic nucleus, or the MMPO.
But the MMPO isn't a thermometer. It doesn't know the temperature on its own.
No, it has to rely on a massive distributed array of sensors all over the body.
I really want to know how the body actually detects hot and cold at a molecular level. It's not magic. It's all about these proteins called TRP channels, right? Transient receptor potential channels.
Yes. TRP channels are a marvel of evolutionary engineering. These are specialized ion channels located at the endings of sensory neurons in your skin, deep in your viscera, and even directly within the brain tissue itself.
And what makes them special?
They're thermosensitive, meaning they literally change their physical three-dimensional shape in response to different ambient temperature ranges.
So heat physically warps the protein.
It alters the conformation of the channel. For instance, TRPM8 is the specific channel that mediates cold sensing.
TRPM8, okay.
When the temperature around the nerve ending drops, the TRPM8 channel physically opens up. This allows positively charged ions, calcium and sodium, to flow into the neuron, depolarizing it, and generating an electrical signal that fires up the spinal cord saying, the environment is getting cold.
Again for heat.
Conversely, TRPM2 channels and several others mediate warm sensing. They open when the kinetic energy increases.
Okay, so I walk out into the snow, the TRPM8 channels in my skin pop open, ions rush in, and an electrical signal shoots up my spinal cord and converges on the MNPO in my brain.
Exactly right.
But what happens next? If I am freezing, how does the MNPO actually warm me up?
The MNPO acts as the commander, initiating a highly orchestrated descending autonomic cascade.
Through what pathways?
The neurons in the MMPO project down to the medial preoptic area, which then signals the dorsomedial hypothalamus, which in turn projects all the way down to a region deep in the brainstem called the raphe pallidus.
Let's pause on the raphe pallidus. What is its job in this chain?
It acts as the final, powerful excitatory relay before leaving the brain. The raphe pallidus sends signals straight down the spinal cord to excite those sympathetic preganglionic neurons we talked about earlier.
The ones that control the blood vessels.
Right. These sympathetic neurons then execute the physical changes in the body. First, they cause intense vasoconstriction of the blood vessels in your skin.
Diverting warm blood away from the surface where heat is lost.
Exactly, trapping it deep in your core to protect your organs. They also actively inhibit any sweating. And Crucially, in many mammals and human infants, they stimulate brown adipose tissue, brown fat, to initiate non-shivering thermogenesis.
Brown fat. How does fat generate heat without moving?
It's fascinating. Brown fat is packed with mitochondria that contain a unique protein called uncoupling protein 1.
What does that do?
Normally, mitochondria burn fuel to create ATP, the energy currency of the cell. But in brown fat, the sympathetic nervous system triggers this uncoupling protein, which essentially short-circuits the mitochondria.
Short circuits them.
Yeah, instead of using the energy to make ATP, the energy is completely dissipated as raw heat. It is a biological space heater.
Wow. But if I am standing in the snow long enough, I start shivering. That mechanical, violent, involuntary shaking? How does the hypothalamus force my muscles to do that?
Shivering is controlled through a parallel but distinct descending pathway. Cold signals from the skin eventually activate different excitatory neurons in the raphe pallidus.
Okay, so the different set of wires.
Yes. These send projections down to the ventral horn of the spinal cord, where the motor neurons live. But they don't target the main muscle drivers right away. They target the much smaller gamma motor neurons.
I've always been confused by the motor neuron alphabet soup. What is a gamma motor neuron doing?
Think of it this way. Deep inside your skeletal muscles, you have tiny sensory structures called muscle spindles, and inside those spindles are intrafusal muscle fibers.
Like little sensory strings.
Exactly. The gamma motor neurons cause these tiny intrafusal fibers to contract. When they contract, it physically stretches the sensory part of the spindle.
And that stretch does what?
This mechanical stretch activates a massive reflex arc that feeds right back onto the large, powerful alpha motor neurons, the ones that actually move your limbs.
Oh, I see.
Because the gamma neurons are keeping the system taut, the alpha motor neurons become highly sensitized and begin to undergo rapid rhythmic bursts of electrical activity.
The shivering.
Right. The result is increased overall muscle tone and the rapid involuntary contractions we call shivering. You are essentially using the friction of muscle fibers grinding against each other to generate heat.
I need to ask a conceptual question here because I think a lot of people have the same misconception I did. When we talk about regulating temperature, the immediate intuitive analogy is a thermostat in a house.
Sure, the classic set point model.
Yeah. You set the dial to 72 degrees. If the room drops to 70, a sensor detects the error, sends a signal, and the heater kicks on until the room hits exactly 72 again, at which point it shuts off.
Right, but the biology doesn't work that way.
The textbook actually goes out of its way to dismantle this set point idea. It states that looking for a single group of neurons acting as a master 37 degrees Celsius chip in the brain is outdated and incorrect.
It is a profound shift in how we understand physiological control.
But if there is no central set point, how does a highly precise, life-sustaining system actually function?
Well, the thermostat model is a classic engineering negative feedback loop with a dedicated, singular error detector. The biological brain relies on what we call the settling point model.
The settling point. The text uses an analogy of a lake, which I found fascinating but a bit dense. Can you break that down?
Absolutely. Let's visualize a lake that is fed by a mountain river coming in and drained by a river flowing out.
Okay.
The water level of that lake stays remarkably constant over time. Why? It isn't because there's a mechanical sensor floating on the surface detecting the exact water level and a computer adjusting a dam. It stays constant purely because of the physics of the system. If there is a massive rainstorm, the lake level naturally rises. But as the depth increases, the hydrostatic pressure pushing down increases.
So it pushes more water out?
Yes. This increased pressure mechanically forces more water out through the draining river at a faster rate. Eventually, the increased outflow mathematically matches the increased inflow, and the lake level stabilizes at a new, slightly higher constant.
Oh, wow.
The constant level is an emergent property of the system's physical dynamics, not a program set point.
It finds its own balance based on the forces acting upon it? So applying that to our core body temperature.
Body temperature is the emergent settling point of multiple independent sensor effector loops that don't actually communicate with each other.
Wait, they don't communicate?
Not directly, no. You have one neural loop entirely dedicated to controlling skin blood vessels. You have a completely separate loop controlling brown fat thermogenesis. You have another independent loop controlling shivering. Each of these loops has its own distinct temperature threshold at which it activates, and its own level of sensitivity. When you walk into the cold, all these independent loops engage to different degrees based on the input from those TRPM8 channels.
So they're all just reacting on their own?
Exactly. The apparent set point of 37 degrees Celsius is just the mathematical settling point, where all these independent thermal defense mechanisms balance out the heat loss to the environment.
That's incredible. There is no master thermostat making a conscious decision. There is just a symphony of independent reflexes that collectively and blindly keep you alive.
That's exactly it.
Okay, but what if I get sick? When I get a fever, my body temperature goes up to 101 degrees, and it holds there. I feel freezing cold, I shiver, my body seems to be actively defending a higher temperature.
That's a great example.
If there's no set point dial to turn up, how does a fever work? Does the whole lake just rise?
That is exactly what happens. When you have a bacterial or viral infection, your immune cells release inflammatory cytokines.
Like an immune alarm.
Right. These cytokines travel to the blood vessels in the brain and trigger the production of prostaglandin E2. This prostaglandin binds directly to receptors on the neurons in the preoptic area.
And what does that do to the loops?
It physically changes their firing rate, essentially raising the activation threshold for all those independent loops. It shifts the entire dynamic system upward.
So my body thinks 101 is the new normal.
Exactly. Your body now defends a higher settling point. You shiver at 99 degrees because, to the new system dynamics, 99 degrees is now perceived as a dangerous heat deficit.
It is a beautifully decentralized network of survival, but temperature is just one variable. We have to look at water balance, and thirst.
A critical system.
If I go for a run and sweat out a liter of fluid, or if I sit on the couch and eat an entire bag of salty chips, the osmolality of my blood changes. The ratio of salt to water is thrown off.
The blood gets thicker, more concentrated.
Right. How does the hypothalamus detect and fix this?
It relies on a triad of structures. We already mentioned the MNPO, the median preoptic nucleus, which acts as the integrator for the data.
The command center.
But the MNPO cannot measure blood chemistry itself. It gets its raw data from two absolutely critical sensor regions, the vascular organ of the lamina terminalis, which we call the OVLT, and the subfornical organ, the SFO.
And the anatomical genius of the OVLT and the SFO is where they are located. They sit outside the blood-brain barrier.
This is a crucial evolutionary adaptation. We have to understand why that is absolutely necessary.
Let's lay it out.
The blood-brain barrier is a microscopic fortress. It is a tight layer of endothelial cells that keeps the delicate, highly sensitive electrical environment of the brain strictly separated from the wildly fluctuating chemistry of the blood plasma.
It protects the brain from toxins and pathogens.
Right. But if your specific job is to monitor the exact, real-time chemical composition of the blood, being locked behind that fortress is a massive functional disadvantage.
As I like to think of it, you can't taste the soup if you're locked out of the kitchen.
That's a perfect way to put it. The OVLT and the SFO are what we call circumventricular organs. They are brain structures, but their capillary beds are fenestrated. They have tiny pores, just like the ones we saw in the pituitary.
So they're exposed.
This means the neurons in the OVLT and SFO are exposed directly to the systemic circulation. They can directly taste the blood.
What are they tasting for?
They contain specialized osmoreceptor neurons that physically shrink or swell as the concentration of salt in the blood changes, altering their firing rate.
So they measure the salt directly,
Yeah.
but they also measure hormones, right?
Yes, and this is why being outside the barrier is doubly important. They are the only way the brain can detect a circulating hormone called angiotensin II.
When would angiotensin II show up?
Imagine you suffer a severe hemorrhage or extreme dehydration. Your blood volume plummets. Your kidneys detect this drop in pressure and release an enzyme called renin, which kicks off a cascade in the blood that ultimately leads to a massive spike in angiotensin II.
And the brain needs to know about that.
Desperately. But angiotensin II is a large peptide. It is physically too large to cross the blood-brain barrier.
Ah, so that's why they need to be outside.
Exactly. Because the SFO and OVLT sit outside the barrier, their neurons have receptors that detect this angiotensin II immediately.
So the SFO and OVLT taste the salty, concentrated blood. They detect the panic signal of angiotensin II from the kidneys, and they send an urgent, excitatory signal up to the MNPO.
The signal integration happens there.
And then the MNPO triggers the conscious psychological feeling of profound thirst, driving you to find water, while simultaneously telling the posterior pituitary to dump vasopressin into the blood to command the kidneys to stop making urine.
It's an incredibly direct, elegant chain of command.
It really really is. But as elegant as water and temperature are, the textbook makes it abundantly clear that they pale in comparison to the complexity of energy.
Oh, absolutely. Energy is a different beast entirely.
We have to move into the deep waters now. Energy balance and hunger. I am going to warn you, listener, we are going to do a very extensive breakdown here, because this is by far the most complex homeostatic parameter the brain has to manage.
It is exponentially more complex. Think about the physical realities. Water and heat are immediate. They are managed continuously, in real time.
You can't store up heat for a week.
Right, but energy is episodic. We eat discrete meals, but we expend energy constantly. Therefore, the system has to manage fuel storage, packing it away as fat. It has to mobilize it when we fast. It has to anticipate meals, process digestion.
It's a massive balancing act.
And balance dozens of competing hormonal signals from the gut, the pancreas, and the adipose tissue. The neural epicenter for this massive calculation is the arcuate nuclei, located at the very bottom of the hypothalamus.
I want to break this down logically. Let's start with short-term signals. The day-to-day, hour-to-hour management. You sit down at a restaurant, you eat a meal, and eventually you feel full and stop eating. What are the biological messengers managing that single event?
When you begin to eat, food hits your stomach and enters your small intestine. The physical presence of nutrients triggers the endocrine cells lining your gut to release a suite of peptides into the local tissue.
What kind of peptides?
Cholecystokinin, or CCK, is rapidly released from the intestine. We also have GLP-1, which has become very famous recently in the context of weight loss drugs, and PYY.
And what do these do?
These gut peptides serve as acute satiation signals. They essentially tell the brain, nutrients are arriving in the digestive tract, the deficit is being resolved, you can stop the motor program of eating now.
But how do they get that message from the intestine all the way up to the brain fast enough to stop you from overeating? Do they float through the blood and cross the blood-brain barrier?
They might cross in small amounts, but their primary, fastest route is far more direct and neurological. These peptides bind locally to receptors on the sensory fibers of the vagus nerve.
The vagus nerve again.
It's the massive superhighway, heavily innervating the entire gastrointestinal tract. When CCK or GLP-1 binds, the vagus nerve fires, acting as a high-speed data cable, shooting these satiety signals directly up into the brainstem.
Specifically terminating in the nucleus of the solitary tract, the NTS.
Exactly.
We talked about the NTS in the first half. It's the primary sensory relay for the viscera.
You got it. The NTS receives this vagal barrage of fullness data and relays it upward to the hypothalamus to terminate the meal.
So CCK, GLP-1, PYY, they are the brakes on the system. But what hits the gas? If I haven't eaten all day, what makes my stomach growl?
The main short-term gas pedal identified so far is a hormone called ghrelin. Ghrelin is unique because it is released primarily by the endocrine cells of the stomach itself.
How does it know when to release?
Its secretion follows a circadian rhythm, peaking right before you typically eat when the stomach is entirely empty. It acts as a systemic blood-borne signal that travels straight to the arcuate nucleus to initiate the feeling of hunger and drive food-seeking behavior.
And then it turns off.
As soon as you eat and the stomach stretches, ghrelin levels plummet back down.
Okay, so ghrelin and CCK manage a single meal. But that doesn't explain the long-term picture. How does the brain know your overall energy status?
It needs a bigger gauge.
Right. How does it know if you are fundamentally starving to death in a famine? Or if you are carrying 50 pounds of excess adipose tissue? That requires long-term signaling.
And the two massive players here are insulin, which tracks blood glucose, and leptin.
And the history of leptin is one of the most incredible and ultimately tragic sagas in modern biological science.
It is a defining story of neuroendocrinology. It starts way back in the winter of 1949 at the Jackson Laboratory in Bar Harbor, Maine.
Let's set the scene.
Researchers there were breeding colonies of mice, and they noticed a spontaneous genetic mutation in one specific lineage that produced wildly, massively obese mice.
Like how obese?
They were insatiable. They ate constantly and grew to 3 times the size of a normal mouse. The researchers named the mutation OB for obese. 16 years later, in a completely different lineage, they found another mutation causing the exact same extreme obesity, which they called DB, for diabetes, because those mice also developed severe high blood sugar.
So they have 2 different genetic mutations causing the exact same extreme outcome. How did they figure out what was broken?
They used a technique called parabiosis, which sounds like science fiction.
Really does.
Researchers surgically connected the circulatory systems of a normal mouse within a mutant mouse, sewing them together so they shared a single blood supply.
And what happened?
When they connected an ob mouse to a normal mouse, the ob mouse stopped overeating and lost its excess weight.
Wow.
This suggested the ob mouse was genetically incapable of producing a circulating fullness hormone, but when it received that hormone from the normal mouse's blood, its brain responded and cured the obesity.
But what happened when they did the parabiosis with the DB mice?
This is the crucial part. When they connected a DB mouse to a normal mouse, the DB mouse stayed massively obese, but the normal mouse completely stopped eating and starved to death.
Starved to death. Why?
The conclusion was brilliant. The DB mouse was producing massive amounts of this unknown fullness hormone, but its brain lacked the receptor to hear it. It was deaf to the signal.
Oh,
So the DB mouse pumped out more and more of the hormone into the shared blood supply. The normal mouse's brain, which had normal receptors, was suddenly flooded with this massive fullness signal, tricking it into thinking it was carrying immense amounts of fat, causing it to completely stop eating.
and it took nearly 5 decades, right, until 1994 for Jeffrey Friedman and his team at Rockefeller University to finally clone the ob gene and discover the physical molecule it encodes, leptin.
Finally giving a name to the signal, yes.
Leptin is a hormone produced directly by your adipocytes, your fat cells. The more physical fat you have on your body, the more leptin you secrete into the blood. It travels to the arcuate nucleus and acts as a long-term gauge of your energy reserves.
That discovery sent shockwaves through the scientific and medical communities. The initial, very exciting multi-billion-dollar hypothesis was that leptin was the ultimate cure for human obesity.
The logic seemed flawless, right? Give an obese person leptin injections, their brain finally realizes they have plenty of stored energy, the hunger shuts off, and they lose weight.
But the textbook explains why that didn't work. And this is crucial.
I really want to spend time unpacking the leptin paradox.
We viewed it through the lens of modern medicine, wanting it to be an anti-obesity drug.
What is its actual purpose?
Leptin's dynamic range is wildly asymmetric. Its primary evolutionary job over millions of years is not to prevent an animal from getting fat. In the wild, in an ancestral environment, getting fat is a massive survival advantage. It means you might survive a winter famine.
So it's not trying to keep you thin.
No. Leptin's true primary job is to aggressively defend you against starvation.
Explain how that asymmetry works mechanically in the brain.
Think about the bottom end of the scale. When your fat stores drop dangerously low, leptin levels in the blood plummet. This sheer absence of leptin acts as a screaming biological klaxon in the hypothalamus.
Like an alarm going off.
A massive alarm. It triggers an intense, overwhelming, all-consuming drive to find food. It simultaneously shuts down energy expenditure. You become lethargic. Your body temperature drops slightly.
It even shuts down reproduction, right?
Yes, it halts the reproductive axis because a mammal cannot physiologically afford the immense energy cost of a pregnancy if it is starving.
Yeah.
Leptin is incredibly, overwhelmingly strong at the bottom end of the scale. It defends against an energy deficit with everything it has.
But on the high end, when someone gains excess weight,
When you have excess fat and leptin levels are soaring above normal, the brain's response is remarkably frail. High leptin does produce some effect to restrain eating, but that weak signal is incredibly easily overridden by the presence of highly palatable, calorie-dense modern food, or by social cues, or by stress.
so it just ignores the high signal.
The defense against high energy stores is weak because, evolutionarily, there was rarely a penalty for storing extra energy. The biological system is rigorously rigged to protect against starvation, not against abundance.
So we have the short-term meal signals like ghrelin and CCK, and the long-term fat signals like leptin and insulin. All of this data pours into the arcuate nucleus. Let's walk through the microscopic battle that is constantly raging inside that nucleus.
It is a literal tug of war.
Between two distinct populations of neurons, the hunger promoters and the satiety promoters. Let's start with the dark side, the hunger promoters.
These are the AGRP neurons. They are named after a specific peptide they produce, a Goody-related peptide. These neurons are the engines of hunger.
When do they fire?
When leptin levels are low indicating starvation, or when ghrelin levels are high indicating an empty stomach, these AGRP neurons are disinhibited and begin to fire rapidly.
And how do they enforce that feeling of hunger on the rest of the brain?
They utilize a triple threat of chemical messengers to aggressively inhibit downstream pathways. First, they release GABA, the classic fast-acting inhibitory neurotransmitter. Second, they release neuropeptide Y, or NPY, which is a potent appetite stimulant.
And the third.
Third, they release the AGRP peptide itself. All three of these molecules target and inhibit a specific set of second-order neurons located downstream in the paraventricular nucleus, the PVH.
And what do these target neurons have?
They express a receptor called the melanocortin 4 receptor, or MC4R.
So the AGRP neurons bombard the MC4R neurons in the PVH and shut them down. What happens when those MC4R neurons go silent?
You feel ravenous. You become obsessed with finding calories. The inhibition of those MC4R neurons is the neurological equivalent of throwing open the floodgates of hunger.
Now, on the other side of this microscopic tug-of-war, we have the satiety promoters, the neurons trying to stop you from eating.
Right. These are the POMC neurons, named for a large precursor protein they make called propiommelanocortin. They work alongside another group of neurons that release glutamate, the VGLUT2 neurons. When leptin levels are high, indicating you have plenty of fat reserves, or when CCK signals a full stomach, these POMC neurons are activated.
Wait, let me pause you. VGLUT2 neurons, what does that mean? Let's not lose anyone in the alphabet soup.
Apologies. VGLUT2 stands for vesicular glutamate transporter 2. It simply identifies a population of neurons that use glutamate, the brain's primary excitatory neurotransmitter, to quickly activate their targets, working in tandem with the slower peptide signals from the POMC neurons.
Got it. So the POMC and VGLUT2 neurons are firing. How do they fight back against the AGRP hunger signals?
The POMC neurons process their large precursor protein into a smaller active peptide called alpha-MSH. Alpha-MSH travels down to those exact same MC4R receptors in the PVH.
But instead of inhibiting them like AGRP did.
Alpha-MSH binds to the receptor and turns it on. It activates the MC4R neurons. When those neurons fire, the motor program for eating is suppressed. You feel full, you feel satiety.
So it is a literal chemical battle for physical control of that single MC4R receptor.
Absolutely.
The POMC neurons are trying to plug alpha-MSH into the receptor to turn on satiety. Meanwhile, the AGRP neurons are actively fighting them, releasing the AGRP peptide, which acts as an antagonist.
It physically jams itself into the receptor to block alpha-MSH from binding,
while simultaneously suppressing the neuron's electrical activity with GABA. Whoever wins that microscopic battle at the MC4R receptor decides whether you binge eat or push the plate away.
That is precisely the mechanism. It is a highly localized, fiercely competitive, high-stakes molecular competition determining your behavior.
Which brings us to what I thought was a truly profound philosophical and behavioral implication buried in the text. This isn't just about moving molecules around to balance an energy equation.
It goes much deeper than that.
It's about the conscious, subjective experience of being alive. The textbook introduces the drive reduction model of hunger. This radically reframes how we traditionally think about wanting food.
It does, and it changes how we view motivation itself. The drive reduction model suggests that hunger is not inherently a positive pursuit of pleasure.
We think we eat because food tastes good.
But the model argues that hunger is inherently an aversive, deeply unpleasant state. When you are fasting, those AGRP neurons become highly active. And clever experiments using optogenetics, where researchers can artificially force AGRP neurons to fire using light, have shown that stimulating AGRP neurons in an animal is deeply aversive.
How so?
The animal will work incredibly hard, pressing levers or avoiding certain areas just to turn those neurons off.
So the act of eating when you are truly biologically hungry is not just about acquiring something good. It is primarily about escaping something terrible.
Exactly.
Eating is simply the relief of this unpleasant, deficiency-induced state of suffering that the AGRP neurons are forcing upon your consciousness.
Precisely. The rapid reduction in AGRP neural activity when you finally eat, or remarkably, even when you just see food and anticipate eating, provides a powerful, rewarding relief.
It feels good to stop the pain.
Right. It functions as a foundational teaching signal. It trains the brain's reward circuitry to constantly seek out food in order to alleviate the biological suffering of starvation.
This explains so much about human behavior. It explains why restrictive dieting is biologically excruciating.
It's an uphill battle.
When you go on a crash diet, you aren't just making a cognitive choice to eat fewer calories. You are intentionally triggering a massive drop in circulating leptin, which removes the brakes and supercharges your AGRP neurons.
You're sounding the alarm.
You are forcing your brain into a state of perceived starvation. You are fighting an intense, hardwired, deeply aversive signal that is screaming at your cortex that you are dying. Human willpower is just a tiny, newly evolved cortical mechanism trying to fight a multi-million-year-old survival algorithm. It's no wonder the diet usually fails.
That is the tragic reality of our evolutionary biology colliding with a modern environment of endless food abundance. The system was built flawlessly for a world of extreme scarcity, and it breaks down when exposed to a drive-through window.
Wow. Let's shift away from the individual for a moment. So far, we've discussed how the hypothalamus vigorously defends its internal energy, water, and heat.
Keeping the host alive.
But survival isn't just about the individual. It's about passing those genes on. And that requires behaviors that go beyond simple homeostasis. We are moving into the sexually dimorphic regions, areas that look or function differently in males and females.
These are critical circuits.
These circuits control sex, aggression, and parenting. They don't respond to a drop in blood sugar. They respond to social cues in the environment and internal gonadal steroids like estrogen and testosterone.
Right. We are shifting from managing the internal milieu to navigating the external social world. And the core anatomical hubs for these social behaviors are located in two specific hypothalamic regions, the preoptic area, or POA, and the ventrolateral aspect of the ventromedial hypothalamic nucleus.
Which is a mouthful, so we refer to it as the VLVMH.
Yes, the VLVMH.
And these areas don't operate in a vacuum. They are heavily interconnected with limbic structures. The text mentions the medial amygdala, the MIA, and the BNSTMPM. I need you to translate BNSTMPM before we go further.
The BNST stands for the bed nucleus of the stria terminalis. The MPM just denotes a specific subregion, the medial posteromedial part. The critical thing to know is that the medial amygdala and the BNST are heavily involved in processing chemosensory cues, specifically pheromones.
Like smells.
Exactly. When an animal smells another animal, that social and reproductive data is processed here and fed directly into the POA and the VLVMH.
And what makes the neurons in the POA and VLVMH special?
Crucially, they uniquely express high levels of sex hormone receptors for estrogen, progesterone, and androgens. Furthermore, many of these specific neurons express an enzyme called aromatase.
What does aromatase do?
Aromatase takes circulating testosterone from the blood and converts it directly into estrogen right there within the local brain tissue. This allows gonadal steroids to fundamentally alter the excitability of these circuits. The hormones prime the circuits, loading the behavioral software, making them ready to fire when the right environmental cue, like the pheromone of arrival or a mate, arrives.
I want to focus deeply on the VLVMH because its role in sex and aggression is what I referenced at the very beginning of the deep dive with the rubber glove. It acts as a dual control switch, particularly in males.
The optogenetic experiments targeting the VLVMH are some of the most revealing in modern behavioral neuroscience. Researchers isolated the specific neurons in the VLVMH of male mice that express the estrogen receptor.
Okay.
They inserted light-sensitive ion channels into these specific neurons, allowing them to control the firing rate of the neurons with a laser pulse delivered through a fiber optic cable in the skull.
Ah, the intensity dial experiment.
Yes. They discovered that the behavior produced depends entirely on the level of activation. If they apply low levels of optogenetic stimulation, just a mild rhythmic activation of these VLVMH neurons, the male mouse initiates mating behavior.
If a female is present.
Exactly. It will attempt to mount her.
Mm.
The low signal basically says reproduce.
But then they turn the dial up.
When they dial up the laser intensity, driving the neurons to fire at a much higher frequency, the behavior instantly radically switches. The mouse goes from mating to intense, indiscriminate aggression.
Indiscriminate.
It will violently attack an intruder male. But it goes far beyond normal territorial defense. Normally, a male mouse relies on pheromones to identify a threat. It won't attack a castrated male, and it certainly won't attack a female.
Because they don't smell like a threat.
Right. But under high VLVMH optogenetic stimulation, the hardwired motor program for aggression is completely forced open. They will brutally attack castrated males. They will attack females.
And they will attack an inflated rubber glove dropped into the cage. That visual is stunning to me. It proves that the aggression isn't some reasoned cognitive decision based on an actual threat.
The mouse isn't angry at the glove.
Right. It is a hardwired robotic motor program waiting to be unleashed by these specific neurons. Low stimulation equals mate. High stimulation equals destroy.
It beautifully and terrifyingly illustrates how these ancient, complex survival behaviors are physically encoded in distinct, overlapping neural populations in the hypothalamus. The behavior is just an output of the circuit.
And the story gets even wilder when we look at parenting behavior, which is controlled by the other main region, the preoptic area, the POA.
Parenting requires a completely different suite of behaviors. This is especially true in males of many species, including mice, which are naturally hostile and infanticidal toward pups that are not their own.
Meaning they kill them.
Yes, a virgin male mouse will typically attack and kill strange pups. But the POA contains a specific subset of neurons that express the peptide galanin.
And these galanin-expressing neurons are functionally distinct from the mating and aggression neurons we just talked about.
Completely distinct. Researchers performed a similar optogenetic experiment. They took a virgin male mouse, a male whose default biological programming says, kill that unrelated pup. They dropped a pup in the cage, but simultaneously turned on the laser to selectively stimulate just these galanin-positive neurons in the POA.
And what happened?
The aggression was completely abolished, instantly. The male dropped all hostility and immediately began engaging in intricate, nurturing, maternal-like behavior. It started grooming the pup, licking it, and then huddling over it to keep it warm.
I compare this to a software patch on a computer. You have a piece of hardware, the male mouse, whose default operating system has a line of code that says, if you see a strange pup, execute the kill program.
A very grim program, yes.
But activating those galanin neurons applies a live, overriding software patch that completely overwrites that default response, substituting it with a complex nurturing program. It demonstrates how profoundly the hypothalamus dictates not just our internal organ function, but our perception of others and our outward physical actions toward them.
It suggests that the biological capacity for tremendous violence and tremendous care are wired right next to each other in the brain. And the hypothalamus acts as the switchboard operator, deciding which circuit gets power based on hormonal states and sensory input.
Wow. Okay, we are coming to the end of this massive two-part lecture on Chapter 41. We've covered an immense amount of ground. Let's do a rapid synthesis of the highlights. When we pull back from all the acronyms and specific brain regions, what is the grand architecture we've actually uncovered?
If we synthesize the core takeaways, we have to start with the sheer elegance of feedback loops and settling points. The textbook destroys the idea that the brain is a micromanager rigidly defending a single set point.
The lake analogy.
Right. Instead, it utilizes multiple independent sensor effector loops to maintain a dynamic physical balance for body temperature, fluid balance, and blood pressure. It is a decentralized network.
We've also mapped the dual effector arms of the hypothalamus. It has immediate high-speed electrical control via the descending autonomic networks, the PAG and the MPFC overriding the factory floor to change blood flow in a split second.
The fiber optic cable.
Exactly. And it has broad sweeping chemical control via the neuroendocrine broadcast system, using the posterior pituitary for a direct hormone dump, and the anterior pituitary portal system as a massive hormonal amplifier.
And we see the strict division yet deep cooperation of the autonomic system itself. The sympathetic system aggressively ramping things up for defense and exertion, the parasympathetic system calming things down for digestion and repair, and the enteric system managing the complex environment of the gut.
I just want to ensure you, the listener, grasp the absolute magnitude of the system. Think about everything we have detailed over the last hour. The TRPM8 channels physically warping in the cold to send a signal.
The fenestrated capillaries of the OVLT tasting the salt in your blood.
The AGRP neurons battling the POMC neurons in a chemical tug-of-war over your fat stores. The releasing factors cascading down microscopic portal veins to trigger your adrenal glands. This massive, staggering, unfathomably complex biological machinery is all working silently in the background right now in your head.
It's humbling.
It is keeping you alive second by second without requiring a single conscious thought or effort from you. It is the ultimate biological autopilot.
It is the foundational biological machinery upon which all of our higher-order thoughts, our philosophy, our art, and our experiences rest. If the hypothalamus fails, the cortex dies. But that connection between the basic machinery and our higher-order experience leads us to a final, lingering concept, something the source text explicitly highlights at the very end as one of the great unsolved mysteries of modern neuroscience.
We love an unsolved mystery. What is the gap in our knowledge?
We have spent all this time detailing exactly how the hypothalamus detects a physical deficiency. We know the exact molecular receptors that detect a drop in leptin. We know the exact circumventricular organs that detect a spike in blood osmolality. We know the hardwired circuits.
Okay, so we know the plumbing.
But how does this highly specific, microscopic biological data get translated into the conscious, generalized psychological experience of desire?
The actual subjective feeling of wanting something?
Exactly. The text points out that how this specific homeostatic information flows from the strictly regulated unconscious hypothalamus up into the nonspecific reward, emotion, and perception pathways in the cortex remains a mystery.
It's a huge leap.
How does the electrical firing of an AGRP neuron, which is just a physical cell releasing peptides, suddenly blossom into the conscious, tormenting thought, I really, really want a cheeseburger right now?
Or a glass of water?
Solving that gap. Understanding exactly how a biological deficit transmutes into psychological desire could be the key to understanding and perhaps finally curing human addiction, because drugs of abuse hijack these exact same pathways.
That is a profound thought to end on. The mysterious gap between cellular biology and human consciousness. Thank you for taking this intense two-part deep dive with us into the hypothalamus. We hope it changed how you view the machinery keeping you alive. Keep questioning and keep exploring. Until next time.